Welcome back to Invodent embryology sessions. Isn't it interesting to know how our actually cells are dividing? How are actually the chromosomes that we have discussed in the last session, how are these chromosomes dividing? How is actually a single cell is dividing to two daughter cells? That particular process of a single cell giving rise to two daughter cells with equal distribution of the number of chromosomes to its two daughter cells is known as mitosis. So in mitosis, there is a single mother cell which is dividing into two daughter cells and sharing its chromosome numbers equally to the two daughter cells. Let us go into deeper and understand how actually this mitosis is there and what are several stages in cell division by the process of mitosis. It starts with early interphase. The first thing to learn is about early interphase. In early interphase, as we have discussed in our previous session, there are chromosomes, the chromatin material, which is very much uncoiled and dispersed within the cell. So we cannot visualize the actual structure of the chromosome in the early interphase. What happens in the late interphase? In the late interphase, there is the duplication of DNA material in each chromosome. So in the early interphase, there are two chromosomes, each have only single chromatid. When it comes to the late interphase, the DNA of each chromosome will divide to give rise to the other strand of the chromosome. So in the late interphase, we can witness two strands of the chromosome because of DNA duplication. So this is about early interphase and late interphase. Then what happens? This uncoiled chromatin material starts coiling it up. So that coiled condensed chromatin material will appear as rod shaped. The rod like structures of the chromatin is seen in early prophase. So in early prophase, the uncoiled chromatin material starts coiling and get condensed to form like a rod shaped structures. So that is seen in early prophase. Then what happens is in late prophase, due to a complete condensation of the chromatin material, we can appreciate the exact chromosome structure in late prophase. So as you can see in this picture, there are one chromatid and second chromatid. Two chromatids which are present, and there are two distinct chromosome structures we can appreciate, and we can also appreciate that they are attached at a point called centromere. So this distinguished structure of chromosome can be seen in the late prophase. So when this is happening with the chromosomal structures during the cell division, there are a few other things happening on the other side. What are they are about the centrioles? The two centrioles of the cell are very close to each other in the early interphase. In the early interphase, the two centrioles are very close to each other. When it comes to the late prophase, they start going away from each other. See, they are very close here, and in the early prophase, they started going away from each other, and in the late prophase, they are completely away from each other. So the centrioles have become away from each other, and the chromosomal structures are very much appreciable in late prophase. The other important point to be discussed is that you can see that the nuclear membrane which is present in the early prophase is not there in the late prophase. So the nuclear membrane gets disappeared in the late prophase. Then comes your metaphase. What happens in the metaphase is that the centrioles which are present on either poles will start creating the spindles. These spindles will get attached to the chromatids of each chromosome at the centromere. So at centromere, these spindles which are given by the centrioles will start getting attached. And what happens in the anaphase is that what happens in the Anaphase is that when these spindles getting attached to the centriole, these start splitting. This longitudinal split of the chromatids of individual chromosomes is taking place in the Anaphase. So in metaphase, the centrioles will give us strong spindle fibers to each chromosomes, and individual chromatid will be attached to the spindles at the centromere of individual chromatid, and there will be a longitudinal split. You can see this, for example, this chromosome, this is split like this. This is going this way to this centriole and this is going this way to this centriole. And in this also, this chromosome is split, this chromatid is going to this centriole and this chromatid is coming to this centriole. In this way, there is a longitudinal split towards the centrioles in the Anaphase. Then what happens in the telophase? In the telophase, there is the separation of two daughter nuclei, which you can see here, and there is the reappearance of the nuclear membrane around the individual daughter nuclei, and you can also appreciate the two centrioles are also getting divided, and when cytoplasm gets divided, there are two individual daughter cells that have come up from the single mother cell. This process is known as mitosis. Hope you have enjoyed the session. Stay tuned to Invodent embryology sessions. Keep learning dentistry. Stay super motivated. Thank you.